So the big question everybody has is why did we need to learn about this JMol? What's the whole point of this? Okay. So I had you guys the last 2.5 weeks look at different protein structures and come up with ways to model them. Now, we were still in school. What you would have done is taken one of these simple models and made an actual 3D model out of it using tubers. But unfortunately, we don't have that opportunity, so we can't do that. Okay. But the purpose of JMol is so that you can visualize what proteins look like. Okay. So what's important about it is it allows us to see what proteins look like, and we can customize the images so we can create our own informative models. There are parts of the protein that we may interpret as being important in its catalytic function, if it's an enzyme, the active site, if it's a receptor protein, the binding site. And what we can do is color them and highlight them in unique and customizable ways. And then we can actually print physical 3D models from JMol using those colors and patterns. And then we can figure out what the different structures look like. We can learn about intricacies in the proteins and compare similar proteins to each other. Okay, so that's the whole purpose of JMol. Okay, and it's one of many programs that can be used to visualize what a protein looks like. Okay, so where do these structures come from? Well, you can see that they were coming from the PDB website, right? Or Proteopedia, which actually gets the information from the PDB website, right? That protein database is a database of structures of proteins that were determined from either X-ray crystallography or another process called nuclear magnetic resonance imaging, okay, known as NMR, okay? NMR studies are a little more precise. They give you a little bit better resolution in some cases. Okay, but basically, what scientists did is they made crystals of proteins using various solutions and concentrations. And some of those solutions will trigger something like this to happen in these first two images where you don't get much. So then they go back to the drawing board and they try again. They try different combinations, right? They try different concentrations of protein with salts and ions, and sometimes they put metals in there. And sometimes it works. All right. A lot of it is trial and error. When it works, they get crystals that can form these long structures like this. These are called rods. Or they can get these hexagonal looking like crystals here. And sometimes you get real tiny ones, such as what's shown in here. Those are crystals, they're just real tiny. Okay. But once those crystals are formed in those solutions, we can then collect them and harvest them. Okay. And so first, the proteins are dissolved in various solutions, right? And they grow for a period of time. Some proteins can grow and form crystals within hours. I've seen them grow within 20 minutes. Sometimes it takes days, sometimes it takes weeks. But the proteins are monitored. Researchers will, consistently look at their proteins. You'd never hold your protein tray this way, sideways. This is just one of those old stock images. But the idea is that you monitor them, and then when they look like they have grown to a decent size and they're visible and they have what's called polarization. I'll go back real quick. You see this image here, um, the third image, fourth image right here. This one, you see these different colors? That's not what those crystals look like necessarily. They're polarized. So what we do is we put them under diffraction grading, okay? Kind of like the same thing what's in our sunglasses, and they polarize the light. If you've ever worn polar sunglasses and tried to look at your cell phone, you notice that if you turn your head one way or the other, you can see the screen and the screen disappears, right? And that's because you've got a diffraction grating in the eyeglasses, which polarizes the light and it prevents certain wavelengths from getting through. Okay, so we do the same thing with protein crystals so we can determine whether or not we have protein. Proteins will diffract and polarize, salt doesn't. So some of these crystals here that are clear in this image are probably salt crystals, okay? Because it turns out that a lot of these solutions involve using salts to trigger the proteins to form, okay? And so after the proteins are formed, we collect them and harvest them from the tray. And when we do this, we remove them with a special wand that has a little microscopic loop on the end. That loop basically has a little piece of monofilament, excuse me. And what happens is if you look in this image, you'll see over here there's a little plastic tray. These are what the loops are on. The loops are attached to a magnetic cap, and then the loop comes off on the spine. And what we do is we swoop up the protein with the loop. The loop contains the crystals, and then we have to preserve the protein. So we put them in bubbling liquid nitrogen. Okay, so this is my hand. I believe this was me doing this, putting the protein in the liquid nitrogen first to let it cool down, and then we put it in what's called a puck. Okay, and this puck contains 16 holes for 16 protein samples to go in. Okay, that puck is then stored in liquid nitrogen and brought over to the National Synchrotron Light Source. Okay, once we do that, we can take those proteins that are frozen and expose them to X-rays at the beamline. Okay, and so we're going to collect structural data at the beamline. And then the X-ray diffraction pattern is actually collected from this specific pattern. So we can see here's our loop up close and personal, and then here's our crystal. Okay, and when we shoot that crystal with X-rays, we get a pattern that looks like this. Okay, so this is the result of X-rays hitting the protein crystal and diffracting. The pattern gets analyzed by the computer programs at the beamline, which is then used to make a structural model. Okay, so we went from that crystal, we scooped it up, we then froze it, then we exposed it to X-rays, and we have machinery and computers that take in that X-ray diffraction data, and we're able to produce an image. Okay. Now the computers will take these little dots, right, which represent the electrons and their intensities. Okay, and an X-ray crystallographer can actually look at this and determine what the exact um, pattern is that we have here, and that tells us what kind of protein we're looking at. But we can take that pattern that goes into, as I said, it goes into a computer, and we can start to analyze the structure. Okay, and so this image shows a structural analysis from the beamline. Okay, and so what we're actually looking at here are the physical protein backbone, right? This is the carbon nitrogen backbone of the protein, and all these little circles here, these little, it looks like nets, these represent electron clouds. These represent the cloud of electrons surrounding each atom. And this takes hours to do, but essentially what we're able to do is look at what we've gotten from the X-ray crystallography data and determine a structure. Okay, we start to model a structure. So after several hours of modeling a structure, this information can reveal to us what a protein actually looks like. And so what we get is a fully refined structure. In this case, this is lysozyme. Okay, and so from that data, we're able to look at its structure, analyze it, and make predictions about what's going on with it. Okay, because in different solutions, this protein may have one structure, and then if you add something else to it, such as a salt or an ion, or maybe some kind of compound that could inhibit its function, it might have a different structure. So this is what's going on right now with COVID-19. They're analyzing the structure of what's called the spike proteins on the outside of the virus and trying to determine how the virus interacts with different receptors. They're finding out that it has a very good interaction with the respiratory tract, but also the digestive tract. They're finding that the receptors actually pair well with receptors in the intestines. And that's why they believe that some people that had COVID-19 didn't have respiratory symptoms, but they actually had lower GI issues, and that they had intestinal problems such as diarrhea and bloating and gas. Okay, but that would not normally be associated with it. Some people thought they may have just had a stomach virus. And so there's a lot of research going into how the virus is interacting, and also once they know what these spike proteins look like, that can help them develop a vaccine. Okay, and so that's the big picture of where this all goes. Okay, so the images that you guys have been looking at in JMol all came from the analysis and structural modeling of X-ray diffraction. And that X-ray diffraction came from proteins that were literally shot with X-rays, and those X-rays created that problem, that pattern, right? And then these protein crystals that we're shooting from the X-rays were grown in trays and harvested, okay? And this is what they initially looked like when we were pulling those proteins out, okay? And what that gets you to is the structures that we can look at in JMol. So each one of these protein structures that you guys looked at, all were developed from crystallization, followed by X-ray diffraction and data analysis. And then ultimately, each of these structures was published to the Protein Data Bank so that scientists from around the world could analyze these structures, look at them, make predictions, and maybe come up with different ways to treat different genetic diseases or diseases in general that affect the body. Okay, so that's the reasoning behind JMol. Moving forward, we're going to learn a little bit about the synchrotron light source and the background information on protein crystallography in the next few weeks to give you guys a better sense of what's going on with that. Okay, and that's it. So I hope this was helpful. Keep working on those structures, and again, on Schoology, you can upload those images. I found a nice link from Miss Arakri. She was really nice at putting that together for us so we can figure out how to put these structures up on Schoology. Okay, so have a great week, guys. Stay healthy, stay safe. Talk to you soon.